Deer at the Auxin conference a few years ago to organize a meeting specialized on tropism, which I think or hope that you all enjoy, because I think the focus of the meeting makes almost every talk interesting. And I hope I can be in line with this idea. So I come from Munich, and Munich is famous for the Oktoberfest and for beer. And I'm actually even closer to the beer because the place where I am, Weihenstephan, is supposed to be the birthplace of beer brewing in the world. And wherever you go in the world, you will find beer brewers that have been trained there. And you cannot imagine how big my disappointment was on Sunday after 12 hours on the plane, on the train, to see that. So you don't have to go to Munich, but it's sufficient to go to Nagoya to be at the Oktoberfest. And they will sell actually the beer that is brewed on my campus, less than a kilometer away from my lab. So I hope you find the time to enjoy. Here are the dates. It has started already. So in my lab over the last 15 years, we have been working on HCC1 kinases. And these HCC1 kinases, they belong to a larger family of protein kinases. And the founding member of this protein family is actually phototropin. And as many of you will know, phototropins, they play a role in flowering plants, but they already are present in algae. And amongst this entire protein family, phototropins are the only members that are present in algae. And I will tell you now what I know about the kinases that we are working on, which are not the phototropins. And in the end, I'll give you a little bit of an idea why I think, or we think, that these have specifically evolved in plant plants. So these HCC kinases, phototropins, and the ones that I'm going to talk about, they have homologous proteins in animals. And in animals, these proteins have a very similar structure. So they have a typical serine-threonine kinase domain, which is interrupted by a short fragment here in the animal protein. That's a human protein kinase. And also in animals, they have various members that belong to this protein family. And any protein that you see here on the scheme that is shown in red belongs to this protein family of HCC kinases. And you can see that sometimes they have multiple phosphorylation targets as protein kinases. Very early in development, this HCC kinase that is shown here plays a role in organizing the polarity of the cell as a regulator of the partitioning proteins. So the activity of this protein kinase C defines the anterior and the posterior part of this cell. And this is a function that we believe, and we'll soon be showing, but I will not show it here because it doesn't fit to the context of this conference. These proteins may also have in animal development, plant development. So here you see the entire family of these HCC kinases as they exist in Arabidopsis. We are mainly concerned with this HCC1 family, which is the largest family. HCC4 kinases are the two phototropins from land plants. There are a total of 4, shown are only 2 HCC2 kinases, and then there are 4 HCC3 family kinases. So a famous member from this family is pinoid, or VEC1 and VEC2, which are functionally highly redundant and have very similar functions to PIN1, or the very similar phenotypes to PIN1, for example. So amongst these HCC1 kinases, the best studied ones are the D6PK family kinases, and within this D6PK family kinases, D6PK itself. And on the right side, you have an AlphaFold model of how we imagine that this D6PK looks like. So you have the serine-threonine kinase domain, and it's interrupted by a longer middle domain of 60 to 80 amino acids. And the number of conspicuous motifs that we discovered over time and characterized over time in this middle domain. And I'll show you the results from this characterization as the first part of my talk. So that's how the middle domain could look like. I think that's actually a model that Hiromasa made at the time. And you see these different motifs appearing here on this side. But I'll talk about this later. So in evolutionary terms, we've done a phylogenomic analysis And what you see here is the presence and absence of PIN proteins and also of PILS proteins and of these HCC8 kinases. And wherever you see a colorful dot or a strong dot, it means that the protein is present. And we see here, this clade is the one that, or this line column is the one that represents the phototropins. We see that they are already present in algae, but we have to go to land plants, so to the bryophytes, for example, that we see other members of this family appearing. So the HCC2 kinases are these ones, and these ones are the KIPK kinases, and these ones are the D6 kinases. So they appear first in land plants, and they're absent from algae. So typical feature for all of these HCC1 kinases, but not for the HCC3 kinases, for example, is that they're strongly polarly distributed at the plasma membrane of essentially any cell that we're looking at. They have very strong polarity, and they're also highly dynamic. So this transport to and from the plasma membrane can be blocked with BFA, preferably A, and that's what you can see here. And you can see that within a few minutes, so this is now 7-8 minutes after BFA treatment, most of the protein is internalized, and we can also wash it out. And very readily after the washout, the protein gets back to the plasma membrane. So it means that it cycles very quickly to and from the plasma membrane. And on the one side, the polarity is something that they share with the PIN proteins, about which I will talk now very quickly, very soon. But the dynamic of this trafficking is much higher than what the PIN proteins are doing. So when we knock out these D6PK proteins, so there are 4 that belong to this core family, we get a number of phenotypes. So we have monocots, we have absence of lateral roots, we have only a slight decrease in root phototropism, we have an absence of lateral root development, we have a shift in the auxin maximum in the root tip. In a quadruple mutant, we get very often a non-differentiated embryo that resembles the GNOME mutant, so the target protein of BFA mutant. And we have a non-phototropic hypocotyl in a triple mutant. So all these phenotypes resemble very much the phenotypes that one observes in different PIN mutants. We also get differentiation defects in the shoots, which wasn't shown on the previous slide. We have different types of tropism defects, so slight gravitropism defect, a strong phototropism defect. This shift in the auxin maximum that has been reported for PIN4, I've shown you on the previous slide. So all this at the long time ago was an indication that the kinases and the pins may interact. And that's actually what we found. So they share the polarity. The pins are phosphorylated by the kinases in vitro, in vivo, at sites that we know very precisely. And together with my colleague Uli Hammes, we established an auxin transport assay in frog oocytes. And we could show that the pins, shown here as pin 3 and pin 1, can be activated by the D6PK phosphorylation, showing that there's a functional relationship between the kinase and the pins that goes beyond the co-localization in auxin transport studies, the co-localization in cell biological analysis. So I'll very briefly talk about the next family member, which is the PAX kinase. So this PAX kinase is like the D6 kinase, is polarly distributed. It's actually very strongly polarly distributed. And this polar distribution is most prominent in protoplasm cells in the root. And in the context of our characterization of this PAX kinase and the phenotypes that we observed in this PAX kinase, we collaborated with the laboratory of Christian Hartke, because due to the presence of this kinase in protoplasm cells, we looked obviously at the cell file and we discovered that there are so-called protoplasm gaps that are typical for the BRX protein that Christian Hartke is very intensively studying. And this and a lot of other circumstantial findings that we had at the time brought us together in a very close collaboration where we discovered for the first time that these PAX kinases are regulated by BRX. And this is an observation or a fact that also extends to other HCC1 kinases. And we found that in the context of this developmental process, the PAX activity towards the pins, which they normally would activate, is inhibited by the BRX interaction. And the consequence would be that this cell does not export auxin. So auxin would accumulate in this cell and trigger some developmental processes required for protofluorine differentiation. But after some time, auxin will have accumulated in these cells, and that leads to an inactivation of BRX, because BRX is known to be an auxin-sensitive protein, and then the activity of PAX is de-repressed, and PAX can phosphorylate the pins, and auxin can leave the cell through the pin transporter. Right. So D6 and BRX, D6PK and PAX, and also many of the other HSC1 kinases, they share motifs in the middle domain. And they are shown here in green, in red, and in yellow, and again here in green, in red, and in yellow. And I will show you quickly what these different domains are doing. So the first one is a repeat of a cysteine followed by a proline separated by 2 or 3 more or less random amino acids. We discovered that all of these HSC1 kinases have a variable number of these repeats. D6PK itself, it has 5 of these repeats. And we found that these are targeted by palmitoylation or S-acylation at least, and help to anchor the protein in the plasma membrane. So when we replace the individual cysteines by serine, then we lose gradually. So when we mutate the 4th or the 5th cysteine, the plasma membrane interaction in protoplasts, but also in real plant cells. We have also transgenic plants. It's only easier to show this for the protoblasts here. And when we mutagenize all 5 cysteines, then we completely lose plasma membrane association. Since also the prolines are conserved, it was also an idea that the prolines may play a role, but in none of the mutants that we made for the prolines, we saw an effect. So these cysteines help the protein to associate with the plasma membrane. And biochemically, we could show that this is a target for S-acylation. That's what these experiments here are showing. You can also inhibit S-acylation by using 2-bromopalmitate, which we have done here, and using increasing concentration of bromopalmitate. You can see that D6, which is polar in this experiment, gradually loses the plasma membrane association and also the polarity. So these experiments here, they will also show you that the palmitylation is not only taking place in the middle domain, so the middle domain alone is palmitylated, that's shown here, but also outside of this middle domain. So there must be other cysteines in the protein that we don't know yet, also palmitylated. But here in the middle domain, that's also a function. So the second motif is a lysine arginine-rich motif. So these are positively charged amino acids, and they help the protein to interact with phospholipids. So this is just shown here on this PIP strip incubation. So the D6PK, when incubated onto a PIP strip, it binds to the phospholipids. So there's an electrostatic interaction between the positively charged polybasic region and the negatively charged phospholipids, which in addition to the palmitylation, regulate the plasma membrane association. So we are analyzing these sequences because we're interested in understanding how the rapid trafficking and the polarity regulation of the D6 kinase is happening. And when interfering with the palmitylation or when interfering with this polybasic motif, we got dissociation from the plasma membrane, but not necessarily loss of the polarity. So we got closer to understanding how the polarity is controlled when we analyzed these 2 serines, serines 310 and 11, that sit adjacent to this polybasic motif. So one could imagine that if they are phosphorylated, then this negative charge will antagonize the positive charge here, and that this may help the protein to lose the affinity for one or the other membrane. So for the plasma membrane, or maybe a membrane that is involved in the trafficking of the protein. So that these 2 serines are important, we found when we replaced them by alanine or by aspartic acid. And so you can see here that the protein kinase, which normally is strictly polar, loses the polarity, indicating that the serine phosphorylation is important. We generated an antibody that should detect the phosphorylated variant of D6PK to understand where in the cell this phosphorylation occurs. And we have had very good experience with PIN phosphosite-specific antibodies, which without exception would detect phosphorylated PIN at the plasma membrane only. And there we were initially quite disappointed that we didn't see with this antibody any signal at the plasma membrane. We found, however, when we did BFA treatment, when the protein is rapidly internalized, that all of a sudden this antibody would pick up the protein. So it seems that it's the internalized protein that is phosphorylated and not the plasma membrane resident protein, as you would expect to see a signal here, indicating that maybe the phosphorylation at the sites is the trigger that takes the protein away from the plasma membrane, in line with the idea that the phosphorylation at these two serines takes the protein away from the plasma membrane. So then in this middle domain, there are not only these two serines, but there are also others. So these are less well conserved, but many of them have been reported to be phosphorylated. They're just not so highly conserved that one would intuitively think that they play a role. But we made a mutation that is a deletion of this SUNMAP region, which starts with the SAN amino acids and finishes with the MAP amino acids. That's why we call it SUNMAP. And this SUNMAP variant is completely apolar. So the protein still gets to the plasma membrane, but it doesn't, is not strictly polarly regulated. So in further studies that I cannot show you here for time reasons, we found that there's an upstream kinase that regulates this phosphorylation of the D6PK at these two interesting serines and also at the non-conserved serines. And that this seems to help the protein kinase to leave the plasma membrane and to drive the trafficking. And if this phosphorylation cannot take place, then D6PK resides for longer at the plasma membrane and it will start to laterally diffuse. And we believe that this is actually the mechanism that leads to the loss of the polarity when these phosphorylation sites are mutated or deleted. So the polarity of this D6 protein kinase is a result of the rapid polar retargeting of the kinase to the plasma membrane. If this process is interrupted, then one gets a lateral diffusion of D6PK, and this leads in all the cases to a loss of auxin transport and the respective loss of kinase activity and PIN activity. Then I will very quickly introduce the KIPK family of this kinase family. So there's 3 members, and there will be a talk from my former postdoc, Yao Xiao, who will talk about the function of these KIPK kinases. Also, these KIPK kinases are polarly distributed in any cells that we are looking at, and Yao will talk quite extensively about it. But the conclusion of his studies is that these kinases are required for the hypocotyl of a germinating seedling in the dark, and the germinating seedling in the dark will grow through the soil. That the hypocotyl is sensitive to touch or to the experience of meeting obstacle along the way during the penetration through the soil. And that's an experiment that usually is quite well perceived in the audience. So what we have done here is we have generated double mutants that show the respective phenotype that I mentioned before. So here, wild type and mutant seedlings have been sown on top of the soil, and you can see that there's an even germination on top of the soil. And then the left experiment here, the seeds were buried about 1 centimeter below the ground. So the elongating hypocotyl had to penetrate the soil, and that works quite efficiently for the wild type seedlings. Almost all of them will penetrate the soil, and they will grow very straight. But these KIPK mutant seedlings, they have a problem in penetration. And if you dig them out from the soil, you see that they overcoil. So they have a problem in responding to the constant touch that they experience during the soil penetration. And then they will overreact, and that results in this overbending. And eventually, many of them will not make it out of the soil. So as I said, Yao Xiao, he will talk about this. And we also have a paper on bioRxiv that is much more comprehensive than what I could show. So finally, I'd like to talk about the kinase, which I like to call patchy. So patchy is a Japanese word, and it means the bicycle patch that you use to fix your punctured bicycle tire. And we call this, I like to call this protein patchy, because as you will see, This protein sits at the subapical region of the root hairs, and it doesn't reside like a ring around the subapical region, but like a patch, and this patch can wander around. And this is actually the work of Hiromasa Shikata. It's not of Yao Xiao, I'm sorry. And has antique status in my lab because it's the nicest story we ever made, and it's not yet published after so many years. So what's this kinase doing? So it's specifically expressed in root hairs and in pollen tubes, so in polarly growing cells. It's residing at the subapical region of the tip. And here he made this fantastic movie where you can see the distribution of the kinase while these root hairs are elongating. So what you will see now, I will have the time to play this movie once or twice, is that this kinase sits at the tip, it wanders with the tip, and when the root hair elongation is finished, the kinase, it will hook up with something which looks like the nucleus and move together with the nucleus back to the trichoblast cells that specifically express this protein. So here goes the movie. So you will see a lot of movement. So now you see the protein going back with the nucleus to the trichoblast cell. So what this kinase is doing, it's not very strongly expressed in the native condition, but when we overexpress it, we get the wavy root hair growth. That's shown here. And when Hiro tracked the distribution of the patchy kinase during this wavy growth, he found that the protein accumulates always at the site where the root hair is going to bend. So before the bending actually occurs, the protein will accumulate at the site of the hypocotyl. And he also discovered that when he was analyzing root hairs growing towards each other, that the protein that you can see here, now this root hair will grow when I show you the movie, that this protein will accumulate at the site of contact with the other root hair. So now you see the protein accumulating at the site of interaction. And of course, this could be a mechanism how a root hair, when growing through the soil, can sense its environment and guide the root hair to find its way through the soil particles. And to refine this analysis, Hiro he made a micro device that would allow him to more selectively analyze root hair elongation. And that will be this movie. So when I start this, you will see how this root hair grows against this artificial obstacle. And then you will see that in the bright field, and we'll see the protein distribution. And you will see that the protein goes to the contact site again. More refined skills are needed. So we believe that this kinase is essentially a growth sensor that the root hair uses to direct its growth. And you may, of course, ask what the loss of function mutant is looking like of this kinase. So in a wild-type root hair, it will not grow straight, but it will slightly meander in the same way as a shoot will not grow straight, but slightly meander. And in this kinase mutants, this meandering is lost because the constant reorientation of the root hair to the changing growth vector is lost. So I've shown you about all these kinases because many of them have roles in land plant development. And what is characteristic for land plants is actually, very trivially, that they grow on land and they also have to grow through soil. And I showed you for the KIPK kinases that have role in soil penetration. I showed you for the Apache kinases that have a role in soil penetration of the root hairs. And for these other kinases, I can tell you that they have phenotypes. I showed you in part of them in phototropic response, so in helping the plants to orient their growth on land. So this work was done by many people who have left the lab already. So Hiro is the Apache kinase person, Alina, Ines, and Lana, they worked a lot on the cell biology of the protein. Melina was working on the biochemistry. Yao, he worked on the KIPK kinases, and the work of Pavan I couldn't show you, but we have a poster from my postdoc Namuk Kim, who will present an online poster, and that includes the contributions of Pavan, and Namuk's name is mentioned here. And we collaborate quite intensively with Uli Hammes and with Christian Hartke. And this is my lab at the COVID Oktoberfest party. So thank you very much for your attention, and I'm welcome to take questions.
Thank you, Klaus. Now it's open to question.
Hey, Klaus, thank you very much. Really great to hear about soil. Great to hear about you thinking about how the plant root is exploring it. I was shocked by your root hair movie you showed at the very end. You had a root hair that bifurcated. Is that unusual behavior? Was that wild type?
That's not a shocking root hair. It's a technical artifact because the height of the chamber is a little bit too small, so that the root hair, like a balloon that you may blow up, decides to go both ways.
Okay.
But it's not forbidden to go both ways because it's the plant that's doing it. It's not me.
So what do you think the kinase is doing actually in the root hair? Do you think it's interacting with the cell extension machinery?
Right. So we think that it selectively blocks the transport to this site and helps the root hair to elongate in the other direction.
Okay, so it's essentially affecting deposition of vesicles. Okay, fantastic.
Thank you. I really enjoyed your talk. I was curious about somewhere in the middle, you showed this picture of wild type versus the mutants, where they're sown on top versus they're sown from underneath, and where they managed to, yeah, that picture. So I was just wondering whether you know anything about the mechanical properties of these mutants. Are they any different from the wild type? They maybe be softer or something?
So I guess Yao will show you that these kinases regulate kinesins, ARC family kinesins, and so that has a role in the microtubule skeleton.
Okay, thank you.
Thank you very much, Klaus. Did I understand your phylogeny properly? You think that the ancestor of all of these, or the oldest one of the whole family, is a phototropin?
Yes.
And so all of the other AGCs would have lost the photosensory bit.
Mm-hmm. All right.
Okay.
It's actually the KIPK kinase. It has a little stretch that comes from the phototropin. So this alpha, J alpha part. So that is still there, but it doesn't seem to have a function. We have studied it.
And so, for example, Chlamydomonas, the only member of this family it has is basically the phototropin. That's right. Thank you. Short question.
I'm still blown away by the Apache story, and it's frustrating it's not published yet.
We should publish it, right?
You should do. When, no pressure. So I guess the question is, how does it sense, what mechanoreceptors are being used to sense the touch? What's the relation? I guess it's interacting with a kinase, some form of mechanosensory protein, which then affects this AGC kinase.
This is a very good question. It is really a good question.
And I take it it's a difficult question.
It's not answered. Yeah.
Okay, all right, okay.
Others?
Quick question, Klaus. The kinase that phosphorylates D6PK at serine 3, 10, 11. Do you know the identity of it?
Yeah, should be PTK1.
Ah, okay, thank you.
Okay, it's time to close. We thanks class again. Yes. Next speaker.
Hi, should I share my screen now?
Yeah. No, not yet.
Okay.
You can see your face. Have you started the sharing screen? Oh, okay. Now we can see the PowerPoint.
Okay. And can you that work?
Yeah, well, next speaker is Leah Brandt from the University of Nottingham. So please start it.
Hello, and thank you so much for this opportunity to give this talk today. I'm really enjoying the conference, and I'm just really sorry that I can't be with everyone in Japan. Trying to work out how to move my slides on. Okay. So key to processes such as root gravitropism is communication between these cells here that are sensing the gravity stimulus right at the root tip, and the cells further back, which are the ones adjusting their growth to create the bending response. It's well established that this communication is created by auxin, in particular these directed auxin fluxes through the root's outer layers. So over a series of papers, we've been trying to understand how does cell scale transport create this auxin flux pattern that's really essential for processes such as root gravitropism. To do this, we've been taking a modelling approach, and over the next couple of slides, I just want to explain this approach in a little bit more detail. So for our models, we've been using a multicellular template which is extracted from a confocal image using cell sets. So we use all the real cell geometries and the real cell connectivities. Using this geometrical information, we then create a simulation of different auxin transport processes. So these equations represent passive auxin diffusion across cell membranes, active auxin transport, which is mediated by the auxin carriers, such as the pins and the AUX1, auxin synthesis and degradation, with the level of synthesis depending on the location of auxin biosynthesis enzymes, passive auxin diffusion within the apoplast, as well as auxin delivery from the shoot. Simulating these equations, we are then able to predict the auxin pattern and the auxin flux pattern. So here you see the predicted auxin levels and the predicted auxin fluxes, and consistent with the experimental literature, we see a substantial auxin within the quiescent center region, and then this directed auxin flux pattern with downward fluxes to the center of the root and upward fluxes through the root's outer layers. In order to test these sorts of model predictions, we've been using the D2 Venus auxin sensor. So as well as predicting auxin, we also predict the distribution of D2 Venus. And as I'm sure many of you know, D2 Venus is a fluorescent sensor that is rapidly degraded by auxin through this network of interactions. So this network of interactions is part of our model in order to predict D2 Venus. This then enables us to compare the predicted D2 Venus with what we see in a confocal image. So we've used exactly the same multicellular geometry as from this confocal image, and then we can compare the D2 Venus on a cell-by-cell basis. So this approach has enabled us to understand aspects such as the role of the AUX1 and the role of plasmodesmata. However, in today's talk, I'd like to focus on our latest paper, which is about the ABCBs. So when we started this project, we realized that, the ABCBs were well established as auxin transporters. However, they hadn't been integrated into previous auxin transport models. And I think part of the reason for this may have been because in the experimental literature, it wasn't clear what their role was. There were various different hypotheses, various different scenarios in the experimental literature. And we realized we could take a different approach to this question of how the ABCBs are acting. So while people had used, single cell systems to try and understand how they transport auxin, we could take a different approach, that by integrating these ABCBs into our model, we could understand what effect are they having on the auxin distribution, and try and see if we can then understand which scenario was most likely to be happening actually in the plants. So to do this, we identified 5 different scenarios from the experimental literature. So it's essentially unclear whether the ABCBs are effluxing auxin independently, and the pins are effluxing independently. So this is scenario 1, as you can see on the right-hand side, or whether the ABCBs are actually forming a synergistic component with the pins. So this would be scenario 2, where the ABCBs and pins are entirely acting synergistically. Of course, there may be some combination of the two. So it may be that the ABCBs and pins are both acting independently, but there's an additional synergistic flux where they're both present. Or maybe one's acting independently, as well as this synergistic flux. So we wrote down these 5 different scenarios, and we also integrated the distributions of ABCBs into our model, as you can see here, using the kind of 3 ABCBs that have been shown to be present within the root tip. And the great thing about modelling is it's very easy for us to simulate these different scenarios and see what effect they have on the predicted auxin distribution. So here you can see our predictions with the 5 different scenarios. So these are the predicted D2 Venus distributions. And we see essentially 2 different categories of predictions. So scenarios 1, 3, and 4 both have real contrast between the auxin levels in the outer layers and the inner layers, whereas we have a much more gradual variation in scenarios 2 and 5. Comparing these predictions with D2 Venus, we saw better agreement with the scenarios 1, 3, and 4. The D2 Venus observation suggests there is this kind of quite big difference between the outer and inner layers. So this led us to believe that scenarios 1, 3, and 4 provided better agreement with the wild type auxin distribution. And we see in these 1, 3, and 4, the key thing is that in these scenarios, we have independent ABCB efflux, as you can see here on the right-hand side. That's the key differences between 1, 3, and 4, and 2 and 5. So with independent ABCB efflux, the ABCBs enable auxin to efflux into the apoplast between the adjacent layers, where AUX1 mediates influx into specific tissues. So this ABCB independent efflux appears to be present based on our predictions, and this has a key role in enabling AUX1 to mediate this influx into particular layers. However, at this stage, we were unable to distinguish between these remaining scenarios. But we did wonder whether the different scenarios, we could get more information by looking at knockout mutants. So this slide has quite a lot of figures, but essentially we looked at the single knockouts of the ABCBs and the double knockouts. And we first of all made these model predictions. So each line of figures is the different scenarios, 1, 3, and 4, and each of the columns is a different mutant. As we saw here, that for majority of the mutants, there was no difference between the predictions in the different scenarios. However, one mutant really stood out, and this was the ABCB1, ABCB19 mutant here. And this showed a real difference in the predicted auxin distribution between scenarios 1 and 3 and scenario 4, suggesting this mutant might be worth studying more detail experimentally. And this also seemed like a really interesting mutant to look at because this also has some quite major root phenotypes. So while these model predictions are quite quick to produce, obviously creating lines crossing knockouts with DT Venus takes more time, but now we'd identified with the model which particular mutant we wanted to look at. So here we just see the simulations on the previous slide just to show you them in more detail. The predictions that in the ABCB119 double knockout, we're seeing a real big difference between these different scenarios in our predictions. And then this was the experimental data we collected. And as you can see in the experimental data, we're seeing this really high auxin level in these epidermal cells underlying the lateral root cap, and a much lower levels back here. So these DT Venus observations really agreed much in were in good agreement with this prediction in scenario 4. So this led us to believe that our model is suggesting that scenario 4 is the one that is occurring in plants. So this led us to believe that ABCBs are acting independently, but the pins are predominantly acting through this synergistic component where we get extra efflux where the ABCBs and pins are both present. So suggesting that ABCBs are mediating this codependent efflux. Of course, with modelling, we always have to, produce results with one set of parameter values. And we always need to ensure that our models are robust to that choice of parameter values. So particularly in auxin transport modelling, the key parameters that we really don't have a clear grasp on their numbers are the permeabilities associated with the active transport. So the permeability associated with the pin and the permeability associated with the AUX1. So of course, it was important to check our conclusions against, our choice of these parameter values. For the majority of our parameter values we varied, we saw that again, scenario 4 was actually the one in best agreement between the model and the data. However, if we varied the permeability associated with the synergistic or codependent ABCB pin efflux, we saw that we actually got some good agreement in certain parameter regimes with scenario 3. So just to explain this figure, so F is a metric that represents the agreement between the model predictions and the data. So with small F values, there's better agreement between the model and the data. And if we just kind of focus here on the blue line and the black line, as you see with kind of lower values of the synergistic permeability, which is typically what people are using in the models, scenario 4 is in good agreement, is in best agreement between the model and the data. However, if we move up here to if we move up here to higher levels of the synergistic term, we see that actually scenario 3 becomes the better one. And we do indeed get agreement between the model and the data. So the modelling does reveal that ABCB pin are mediating a codependent efflux. There may be some small component to the independent pin, but it's the codependent efflux that's really the dominant one. So I'm just going to skip over this slide in interest of time. So the other thing we wanted to know was understand one aspect of this is that codependent efflux is essential also to create a significant shootward transport through the tissue. So I'm really sorry, the clock with my timing has literally only disappeared on my screen as I ran out of time. So essentially this part of the project showed that this codependent efflux is also important for the ABCBs to contribute to the directed fluxes. So we see that we only get the ABCBs contributing to directed fluxes in scenarios 2, 3, 4, and 5, the scenarios where we also have this codependent efflux. And this is an agreement with experimental data from Elke Spalding's lab that shows if you knock out ABCB4, you get reduced efflux. So this provides further evidence that this codependent efflux is existing. So in summary, the modelling can reveal how cellular processes create the dynamic hormone distributions. We've shown that ABCBs efflux auxin independently. And the model also suggests that the PIN-mediated auxin efflux is predominantly through the codependent efflux where co-localized with the ABCBs. And I've also shown that the modelling suggests that the codependent ABCB PIN efflux enables the ABCBs to contribute to the directed auxin fluxes, and potentially this explains the gravitropic phenotypes of some of the ABCB knockout mutants. And just to finish, this work was predominantly done by Nathan Meller, who was a modelling postdoc, and Ute Voss, who was doing the experimental work, in collaboration with many other people, predominantly in Nottingham, but also Marcus Geisler, and in particular Malcolm Bennett, who is in the audience with you in Japan. Thank you for listening.
Thank you, Leah, for a nice talk. Question? Class.
Leah, thank you very much for this nice and interesting talk. A few months ago, I believe it was published that ABCB19 is rather a prasinosteroid transporter than an auxin transporter. I mean, although in the paper they didn't negate that ABCB19 is an auxin transporter, they used auxin as a negative control in their experiment. And prasinosteroid was the new cargo discovered of the transporter. How does that fit with your data?
Yeah, I mean, that was a really interesting paper, and that's a really interesting idea that these are actually transporting prasinosteroids. I mean, the modelling at the moment hasn't considered prasinosteroids, but I mean, that would be a really interesting aspect to look at, in the future.
I found the talk very interesting. I just have one thing that I don't know if it's the case, but have you also considered catabolism into your model? Because let's say GH3s, for example, we know that they're going to somehow trap some auxin in specific cells. So how would you implement that into your model, or is it implemented already?
So we have looked at auxin catabolism. We actually published a model with auxin catabolism, I think, maybe 2016. But we found that this didn't seem to have a big effect on the auxin pattern. It seemed that the auxin transporters transport auxin so quickly that it's really the transporters that are creating the auxin pattern. So if you perturb something about auxin, the transporters move back to the same pattern within 10-15 minutes.
Hi, Leah.
Hi, Malcolm.
Thank you for a very nice talk. We had some discussions yesterday after Kirsten's seminar about the role of plasmodesmata in not just radial transport, which is obviously something you discovered, the importance of plasmodesmata in moving auxin across radially in the elongation zone, but also there were questions about its importance moving from the apex shootward, and we asked Kirsten about the relative importance of this transport pathway compared to PIN transport. Do you have any comments or thoughts and reflections about that?
Because the pins are polar, they are creating an auxin velocity, whereas the non-polar components, such as the plasmodesmata, are creating a diffusion. So essentially, if the pins are there, they're always going to be a think they're typically going to be kind of creating a faster auxin stream, a kind of more directed auxin stream. But of course, if the pins are not there, like Kirsten was suggesting in her halotropism, the side by the facing salt, then of course, then the diffusion through plasmodesmata can kind of take over and can allow the auxin to move to different areas of the root tip. So that they're providing an additional way of auxin movement, and of course will be more important in kind of scenarios where the pins are not able to transport auxin so quickly because they've been internalized.
Yeah, looking at the distribution of plasmodesmata, they're very, very dense on the apical and basal sides of cells and less so on the radial sides. I guess, so it's almost like having a leaker bucket, essentially, the pins are there, but you're also leaking auxin through presumably the plasmodesmata are open.
Yeah, and it's an interesting idea that could be particularly important in stress situations or tropisms where, the pins aren't able to act so strongly.
Okay, it's time to move on. Next.
Thank you very much.
Thank you so much. Yeah.
Hey, can you all hear me?
Yeah.
Yeah, can I start sharing my screen?
Yes, please. Yeah, we can.
Can you all see my slides?
Mm-hmm.
Yeah. Can I start?
Yeah, I introduce. Next speaker is Prava Vinishenko from the Center for Plant Microbiology, Tübingen. She will talk about the PIN. I'm sorry.
Yeah.
Mediated by the NPH3, right?
Yeah,
Okay.
exactly. Okay. May I start? Hello?
Yeah.
Can you all hear me, and may I start? Yeah. Okay. Good evening, everyone from Germany. Foremost, I would like to thank the organizers of this amazing conference for giving me this wonderful opportunity to present my work today, which is titled AI-Assisted Decoding of Condensate Assembly in the Plant Phototrophic Response. It is well known that upon unilateral blue light irradiation, plants bend their stem towards the incoming blue light, resulting in the formation of a lateral auxin gradient. On the other hand, the polar localization of the PLIN proteins within the plasma membrane make them likely candidates that are promoting this auxin gradient that precedes the phototrophic growth. On the other hand, in the upstream signaling cascade, we have the blue light photoreceptor FOT1 that is involved in initiating the process of phototrophic response. Downstream of FOT1 is NPH3, which is another plasma membrane associated protein, and it seems to be a very key instrumental factor for auxin redistribution. However, until now, the signaling events that couple all these individual processes remains elusive. Recently, the group of ours and the group of John Christie has identified that blue light induces an immediate phosphorylation of NPH3, resulting in the binding of 1433 proteins, thereby resulting in the dissociation of NPH3 from the plasma membrane into the cytosol, accompanied by dephosphorylation. And most interestingly, this dynamic change in localization is reversible when we transfer NPH3 protein to dark, suggesting that blue light driven and 1433 mediated dynamic change in NPH3 localization seems to be crucial for its function. Considering the NPH3 protein amino acid structure, NPH3 possesses, in addition to the central NPH3 domain, and another BTB and coil-coil protein-protein interaction domains. As you can see here, upon transient expression studies, we clearly see that NPH3 is localized to the plasma membrane in the dark. Upon light, it induces plasma membrane immediate dissociation of NPH3 from the plasma membrane into the cytosol, where it gets accumulated into these discrete particle-like structures. We also identified that 14-3-3 association seems to be essential for this plasma membrane induced light-induced plasma membrane dissociation, wherein the binding mutant incapable of binding to 14-3-3 remains constitutively plasma membrane both under dark and blue light. Furthermore, our group also identified that the plasma membrane association of NPH3 is phospholipid dependent and requires an amphipathic helix in the C-terminal IDR region. And deletion of this C-terminal IDR region resulted in the mutant variant, which I will call now from on delta C, resulted in forming a constitutively discrete particle-like structures, both in dark and light. Furthermore, we also could show that these NPH3 discrete particle-like structures shows the characteristics of a membraneless biomolecular condensates, which are the key driving force behind this formation are multivalent protein-protein interactions. Underlying this phenomenon, our major focus was to uncover all these molecular determinants underlying the formation of cytosolic condensates and to test their functional relevance. To begin with, we performed several co-expression and co-IP analysis to test this. First, we started with co-expressing the constitutively condensate forming variant, here delta C. Upon co-expression, we can clearly see a co-localization of condensates both under dark and light. In addition, the co-IP analysis showed a clear interaction of this variant in the condensates, suggesting that NPH3 is capable of homo-oligomerization. My next objective was to check if the variants that are also capable of plasma membrane association and forming condensates could still co-localize in the condensates. For this, we did a co-expression of NPH3 wild type with that of the delta C, which resulted in an surprising way of reassociation of delta C from the condensates to the plasma membrane, and how surprisingly in the dark, these variants co-localize perfectly in the condensates. And this was further validated by the co-IP analysis, wherein here we can see a strong interaction of these two variants, suggesting that NPH3 self-assembles into high-affinity oligomers at the plasma membrane. And that self-association might be coupled to the dynamic formation of higher-order homo or hetero oligomers. In order to further validate the molecular determinant underlying the self-association, we went ahead by deleting upstream of the region of this thing, which is the coil-coil domain, which is called delta CC. To our surprise, we found that the single expression of this variant was still capable of forming condensates under both dark and light. However, upon co-expression analysis with NPH3, we could show that this does not allow for uniform plasma membrane association of the delta C variant in the dark, but they perfectly showed co-localization in condensates upon blue light. And most interestingly, a co-IP analysis revealed a strong decrease in interaction of delta CC with NPH3, both under dark and light, suggesting a hypothesis that there might exist two types of self-association, one high affinity, another low affinity. We were struggling to decode this phenomenon for quite a while until we came across the most exciting tool, which is the AI-assisted AlphaFold prediction, multimer protein prediction tool. So herein, I could clearly decipher the monomeric structure of NPH3, where here the PLDT value shows a clear characterization of all the defined domains with high confidence intervals. And moreover, the PAE plot also clearly depicts that the relative positions of both the BTP and the NPH3 domain are well defined, while that of the CC, here highlighted in red arrows, is not that clearly defined. So we went ahead with proceeding with the multimer prediction of NPH3. So we, according to the AlphaFold prediction, it suggested that NPH3 is capable of forming a higher order oligomer with the highest probability of trimer formation. And this trimer formation was supposed to be stabilized by the coil-coil superhelices. To our surprise and to our unexpected surprise, we found another 3 helical bundle helices, which is located upstream of the coil-coil, here which is characterized by the linear interacting peptide and short lip, which are also involved in hydrophobic interfaces. And when looking from the above, that is from the cytoplasm to the plasma membrane, you can see that this trimer formation shows a symmetrically arranged 3-bladed propeller. Furthermore, we also went ahead to check the AlphaFold prediction models by deleting the coil-coil, and to our surprise, we still found that NPH3 is still capable of forming a trimer, here highly mediated by the lip motif. Further deletion of the lip completely abolished or did not support our trimer formation, where here this variant is called delta C full, and this variant is incapable of condensate formation, suggesting the phenomenon that there might exhibit a low affinity self-association that might be mediated via the lip. So in order to validate this, we went to co-expression analysis. Here you can see clearly that upon co-expression of delta C with either itself or with the delta CC variant clearly showed a co-localization in the condensates, but a clear separation of delta C full with delta C was observed. And moreover, co-IP analysis showed a strong decrease and stepwise decrease in affinity of interaction, indicating that the coil-coil domain might be involved in a high affinity mediated interaction, while that of the lip domain mediates a low affinity interaction. Based on these results, we propose that NPH3 might contain a unique bipartite C-terminal homooligomerization motif comprising of coil-coil and lip domain mediating self-association in high affinity and low affinity manner, and that lip motif could be sufficient for condensate formation. We went ahead to check if the role of this C-terminal self-association in terms of membrane targeting. It has been already described by us that the amphipathic helix located in the C-terminal region of NPH3 is involved in plasma membrane association, and that it binds directly, the last 51 amino acids directly bind to the phospholipids in vivo. But surprisingly, this was not sufficient for membrane targeting. And to our further astonishing result, inclusion of the coil-coil domain resulted in a clear plasma membrane targeting of this variant, and this was validated by the microsomal fractionation, where you can clearly see a strong enrichment of the protein in the plasma microsomal fraction. And indeed, interruption of this coil-coil resulted in a complete abolition of membrane targeting, which indeed is required for trimer formation. Further, we went ahead to check the role of lip motif in terms of membrane targeting. Here we clearly see that lip motif do not have an apparent effect in membrane targeting. However, interruption in coil-coil domain resulted in a shift in significant amount of protein from the membrane to the soluble fraction. But when you check it at the confocal, this was not obvious. As you can see clearly that these variants looks like they are plasma membrane associated, indicating that the CC district seems to be still lightly attached, very mildly, loosely attached to the plasma membrane, which might be mediated by the low affinity mediated lip motif. Indeed, interruption of both the coil-coil and deletion of the lip clearly resulted in abolition of membrane targeting. These results suggest that the phospholipid binding is required, but not sufficient for membrane association. And in addition, the bipartite C-terminal homooligomerization motif seems to act redundantly in terms of membrane association, in which coil-coil mediates a strong plasma membrane association, while the lip motif is sufficient for weak plasma membrane association. Having already shown that two variants, delta C full and delta lip CC district, that is the bipartite, both the motifs of the bipartite C-terminal homooligomerization directly contributes to oligomeric status of NPH3 condensates, we investigated the direct role of individual component. Here, direct interruption of the CC did not affect the condensate formation or marginally affected, while to our surprise, we found that deletion of lip motif was sufficient to abolish this condensate formation, indicating that lip motif is indispensable for condensate formation, but the CC not. We already have described that the condensate formation requires multivalent protein-protein interaction. So we suspected that might be another potential valence motif that is involved in condensate. Apparently, we ended up in investigating the BTB domain. Here, complete deletion of the BTB domain resulted in a variant wherein here shows lack of condensation, suggesting that the condensation formation requires both the lip motif and the BTB domain. Furthermore, we took the advantage of AlphaFold prediction and checked what's happening with the motif where you delete both the lip and the coil-coil. Indeed, we predicted in a hypothetical way that still NPH3 is able to form a dimer that is mediated by a BTB domain in an anti-parallel BTB-BTB interaction, which is, of course, completely hypothetical. And then upon a closer observation, I would like to emphasize here that the NRL protein family or the NPH3 family proteins contains BTB domain consisting of a core BTB and a C-terminal extension. And here in our case, NPH3 seems to have this antiparallel interaction that is mediated by the alpha 5 of the core BTB domain with the alpha 8 of the C-terminal extension domain of BTB. And we also could end up that just the deletion of the alpha 8 of the C-terminal BTB extension was sufficient enough to abolish the condensation, suggesting that the C-terminal extension of the plant-specific BTB domain is of utmost importance for polymerization of NPH3 in the cytosol. We went ahead to test the functionality of this condensate formation by generating transgenic lines that are incapable of condensate formation. Here we ended up in generating the variants like delta lip and delta BTB alpha 8, where these transgenic lines were incapable of restoring the phenotype of phototrophism, suggesting that NPH3 condensation in the cytosol might be a prerequisite for its proper function. Thus, we could conclude and we could identify that as an overall summary, we identified a unique bipartite C-terminal homooligomerization motif comprising of coil-coil and lip motif that acts redundantly in terms of plasmamembrane association and affinity association and non-redundantly in terms of condensate formation, wherein a C-terminal extension of BTB is also found additionally required for condensate formation. Therefore, with these observations, we ended up in proposing a model, which is a bistable switch model herein, where a conversion of a strict C-terminal trimerization at the plasma membrane into a bivalent cooperative homooligomerization coupled by lip and BTB in the cytosol. With this, I would like to thank all the members of our group, especially my supervisor, Professor Claudia Ocking, for giving me a wonderful opportunity to work under this exciting project, and especially the organizers of this wonderful conference for giving me an opportunity to present my work here. And if you're further interested in my work, you still can access the digital version of my poster, which is poster number 51. And our work is already available as an online in a preprint version. With this, I would like to conclude my talk and thank you all.
Thank you, Prabha.
Yeah.
Question?
Thank you very much, Prabha, for this very dynamic talk. Does your structural analysis, although this is all in silico, offer any suggestions as to how the phosphorylation of this serine, this light-induced phosphorylation by the serine, would induce the internalization of NPH3? Any hypothesis you can propose?
Sorry, come again with your question. How does
the internalization, the light-induced internalization of NPH3 initiated by the phosphorylation of serine 744 or something like that, you know this?
Yes.
And so does your structural analysis offer any hypotheses as to how this initial phosphorylation may trigger this dramatic relocalization?
We actually did a lot of analysis in this regard, and supposedly we couldn't end up in finding something that is associated to phosphorylation, because most of the mutants that we work with shows a perfect initiation and did not show a difference in phosphorylation or the initial phosphorylation status. And as far as of now, we could conclude that the S744 is very crucial for strictly blue light induced phosphorylation, and that is required for 1433 binding. And so far we couldn't really find this is our challenging aspect for the future. This is what we are really trying to answer this. It's a very nice question, but this is our main goal, because we started our project with that focus and we apparently ended in completely a different scenario. Yeah.
Thank you very much.
Yes.
Hi, thank you for your very interesting talk. It was a lot of information, so I'm not sure if I got the last part right. So usually there's this 3-blade propeller structure that you can have.
Yes.
But then there's also this anti-parallel BTB interaction. But is that a dimer? only?
Yes, that's a dimer. And we actually, I did not tell everything because it's a short talk. So this actually, we ended up in the last minute of while we wanted to investigate the role of BTB in homo oligomerization, and we ended up in via in silico that NPH3 is still capable of forming a dimer. But however, if you see in the context of real scenarios, this formation of dimer is highly impossible, because when you see this three-bladed propeller-like structure, you can see that the BTB is inclined on the right-angled propeller. So it's really far away from the plasma membrane. So we really think that it might occur that this is actually results in a formation of a reticulate big spider web kind of things. But in real life or real physiological scenario, this is impossible. So because we still have a coil that is in close vicinity to the plasma membrane and an amphipathic helix and then the lip motif. And when you destroy the lip motif, the right angle propeller structure is completely distorted. And then the BTB domain comes in this close contact. So it's no more, able to form this dimer formation. Yeah.
Sorry, so you're saying that because the interaction with the plasma membrane would be too strong, there would be no chance of the MPH3 only hanging on to other MPH3?
It will be hanging on with other NPH3, but the possibility of hanging through BTB will be really too less. The possibility is too less, because there is a coil, and there is amphipathic helix, which are really acting in a dominant way, and the BTB comes in the very last in this race. So the possibility of BTB in this regard would be really minimal.
Okay, thank you.
Yeah.
Yes, thank you. Okay, short question.
Yeah, so thanks for the talk. So why does the condensation important for MPH3's function? So what does MPH3 do there?
Yeah, it's a good question. Actually, we think that according to a working model, we always think that there should be a proper cycling of NPH3 from the plasma membrane to the cytosol, wherein it forms condensates and recycling back. And this condensate, we presume that it is a site of dynamic sequestration of NPH3 in its proper function, and it should be controlled temporally. So when this condensate formation is either delayed or interrupted, we think that presumably all the downstream signaling events or the upstream signaling events are distorted. So we then think this condensation is really, really important. And the most interesting fact is that we did not identify any other interaction partners of NPH3 in the cytosol except that of 1433. So we really have to think about in a broader aspect, and that's why we also think that there is a lip motif which might be also required and for its function. So we have to investigate about this in more in detail. Yeah. But for now, we think it's very crucial and it's a very prerequisite for proper function.
So you still don't know how does the MPH3 work in the condensates?
Yeah, so we are currently trying some TOBOID approaches to really find its interaction partners. Ultimately, we really are not getting something like that, and we think that it might be resulting in a homotypic interaction, that it just forms homo oligomers in the condensates. However, this is our challenging task. So on one hand, we work on identifying those partners that are involved in early phosphorylation initiating events at the plasma membrane, which is crucial for its dissociation. And secondly, the protein interactions or the involvement of NPH3 condensates in a phototrophic response. So these are two different aspects that really has to work in parallel together, and addressing these questions in different aspects, you have to generate a lot of interesting lines like photoactivable and like condensate controlling mutants, which is really challenging. So yeah, currently this is in progress.
Thanks.
Okay, thank you, Praha.
Yeah, thank you.
Next speaker is Dr. Uri Hamas from Technical University of Munich. Can you hear me? Hi, Uri. Muted. It's mute. Oh.
Can you hear me now?
Yeah.
Okay, fine. And now laser pointer, everything is good to go on my side. Can I start? Hello?
Please start.
Hello? Okay.
Yeah, good.
Yeah? Okay, I'm starting now. Is that fine?
Yes.
Okay, good. So welcome from another talk from Germany. I would have loved to be here, and I thank the organizers for giving me the opportunity to nevertheless present recent data from my group. You all know that auxin is transported by a number of different transporters across the plasma membrane and across internal membranes. And I will be focusing in this presentation today on the pin-formed transporters for the very reason that pin-mediated polar auxin transport is required for gravitropic root growth. It has been well established for decades now that here in the root tip, we have the following scenario, that auxin is transported down in the stele, and then it goes here to the very, very root tip where there is the formation of an auxin maximum and an auxin response maximum. And then auxin is transported back up in the epidermis, mainly by the activity of pin2. And this is the so-called reverse fountain model that gives rise to phototropic growth. What you see here is obviously the typical Columbia wild-type Arabidopsis growing down, and this here is a rather typical, and this is a rather drastical example of a pin2 mutant, because still in pin2, the overall tendency is to grow down. So 2 years ago, we and two other groups were able to solve the structures of pin-formed proteins and to show how auxin is transported. And the movie here is looping all the time, so I can continue talking. What you see here are the two dimers. One is shown in colors and one is shown in white. And what we have is a scaffold domain that is the interaction domain between the two monomers. And then we have the transport domain that is shown here in light green. And now the transporter flips back, and when it's flipped back, auxin is coming in here. It's binding here between the broken helices 4 and 9, which triggers then the rigid body movement of the transport domain, which leads to the dissociation of auxin on the outside. And in all the structures that were determined, the loop structure remained undetermined. I'm going to talk a little bit more and in detail about the loop structure. So for me, when we finally had gotten the structure and understood the mechanism of pins, I thought, okay, fine, I'm done. I can attend to other business. I understand auxin transport, but I was very wrong. So what you see here is a cartoon of the pins. And I'm going to be mainly talking about Arabidopsis and pretend that's reality. So what you see here on the left side are the so-called canonical pins, which is pin 1, 2, 3, 4, and 7 in Arabidopsis. They have 5 membrane helices, and then they have a large intrinsically disordered loop domain, and then again they have 5 transmembrane helices. And the loop here in these protein is around 330 amino acids. So that's about half of the protein size. And all the phosphorylation that Klaus introduced previously occur here in this loop domain in these conserved serine residues. These proteins reside in the plasma membrane, very often in a polar fashion. Then we have the so-called non-canonical pins, and they don't have this large loop domain. They still have a loop domain. However, this is reduced in size. And in angiosperms, we have the situation that we also have pin 6, which is somewhat of an intermediate between the two scenarios. So when you look at a phylogenetic tree of the pins and you look, or you base that on the transmembrane domains, what you find is that pin 3, 4, 7 form a clade that is very, very closely together, and by far and large, it's the same protein. I mean, in terms of their biochemistry, they act genetically redundant and so forth. And then we have pin 1 and 2, the other canonical pins that cluster here with pin 3, 4, and 7. And then we have pin 8 here in the middle, which is a non-canonical pin that doesn't have a long loop. And then here pin 6 and pin 5 are sitting on individual branches. And Tom Bennett has suggested that, and others, that pin 5 probably never had a loop, whereas pin 8 at some point in evolution might have had a loop and then lost it. So we have been working on pin transport regulation together with Klaus Schwechheimer for almost a decade now. And it is by now well established, I would say, that pin transporters are off when they're not phosphorylated by kinases. So due to the action of kinases such as D6PK or Pinoid and a few others, we get the phosphorylation of the loop domain, and this turns the transporter on so auxin can go through. And obviously there must be counteracting phosphatases that can then switch the transporter back off. So the function of the loop is it's auto-inhibiting the pins. And that was further substantiated by the finding that when you look at pin 8 that doesn't have the long loop, this shows constitutive auxin transport that is also kinase independent. And the way we do these experiments is that we use Xenopus levis oocytes, and this is a fairly well-established heterologous expression system. By far and large, all the transporters that have been characterized in detail have been characterized in Xenopus levis oocytes, also by electrophysiology. And what you usually do is you inject the RNA of interest, and then the transporter of interest is made. The big advantage of these oocytes is that they contain yolk. They don't have any endogenous membrane proteins, or by far and large don't have. They have a few, but for nutrients and metabolites, they don't require the uptake, and therefore you can characterize transporters virtually background free in that system. And what we do now for auxin transport is we inject the RNA of the transporter, we co-inject the RNA of an activating kinase, then we wait for 4 days for the proteins to be expressed. And then in a second round of micro-injections, we inject the substrate, which is in our case tritium labeled IAA. And the big advantage of this system is that using micro-injection in these fairly large cells, we can adjust the starting concentration of auxin at the beginning of the experiment, which is a prerequisite if you want to do decent biochemistry, as opposed to passive preloading, where you never know what your starting concentration at the beginning of the experiment is. And these are data of a typical experiment. So shown here on the left, this is the time course. So in this case, it's pin 3. We use 15 minutes. This here is pin 3 without the kinase. Each of these data points is mean and standard error of 10 individually injected oocytes. And as you can see here, for the transporter without the kinase, you don't lose much radioactivity in the time course of the experiment. Now, when you co-inject the RNA of a kinase, what you then see is that at any given time point, the amount of radioactivity in the oocytes is less, which is consistent with auxin transport. And when we now do a linear regression, we can calculate the slope. And when we then plot the slope, this is shown here. So one of these data points here is one slope, such as, for example, this one here. And one of these data points is the slope of one of an experiment like this. So these are biological replicates. And I believe it's very evident that here, without a kinase, we don't have transport. And with a kinase, we have transport. And what we then were interested in was to understand, do these pins have individual properties? Do they have individual kinetics, or do they do the same? Pretty much all the modeling approaches assumed so far that pins have the same kinetics. And what you see here now is an experiment that is done with equal amount of transporter in oocytes. It's normalized to kinase. There is also equal phosphorylation, both in terms of quality and quantity. So what you see is truly the reflection of the transport activity of the protein. And we used two different kinases, D6PK and Pinoid. And what you see here for pin 1 is that in the absence of a kinase at any given concentration, so we went from 0 to 10 micromolar, which we back then thought was the physiological range, with 10 micromolar being almost sublethal. And what you can see here then is that when you co-inject D6PK, you activate transport, so you get more transport. And when you do that same thing with Pinoid, you get more transport. So, and this is for pin 1 and pin 2 the same. We get an asymmetry between the kinase activation. So Pinoid is always working a little bit better. And when you look at that, you can also see that pin 1 is about 2 times as quick as pin 2. Now, when you look at pin 3, there are a couple of things to notice. First of all, this axis here goes to 200, so as opposed to 100, indicating already that pin 3 is much quicker than pin 1 and pin 2. And the other thing that you can see is that the kinase asymmetry, the difference between D6PK and pinoid, is there, but it is not as pronounced as it is in pin 1 and pin 2. So, but what you can see is we have a linear correlation between the auxin concentration and the transport rate. And that was very, very striking to me. And this is why we have been sitting on these data for years. And there may actually be people in the audience who have seen these data before, but now we finally submitted them. The reason why I was so reluctant is, I mean, transporters aren't enzymes, and you cannot apply enzyme kinetics strictly to transporters. What is shown over here is that here is pin. So once IAA has been released, then you have a kinetic component that is the occluded state when it flips back. And then in the inward state, which is again a kinetic component in itself, you also have the kinetic component of substrate binding. And then here, yet another kinetic component, and then you switch back outward. So nevertheless, what can be done and has been done is you can fit Michaelis-Menten models, and you can come up with a half maximal rate that is then also often referred to as Km. There is also another thing, and this kinetic component of substrate binding is called Kd. Nevertheless, I mean, I was really, really worried because I would have expected something like this nonlinear relation, but we got the linear relation. And the answer to the question came when we then characterized pin 8, once we had enough protein and we could reconstitute it in proteoliposomes. This here is a current trace, a couple of current traces from SSM electrophysiology, which I cannot explain in detail, but as you can see, this here is concentration dependent. And when you plot the peak current, which is the interesting parameter in this electrophysiological experiment, what you can see is that you then get a Michaelis-Menten curve. And I want you to notice that we go to 2 millimolar auxin, and I believe we can all agree that this does not happen. I mean, the transporter won't see that. And we get a Km for that of 360 micromolar. The Kd we also established, so the binding of auxin to be to around 40 micromolar. And I also didn't believe these data very much initially, and I thought this is an electrophysiological thing. However, we published it, and at the same time, the 2 other papers on pin 1 and pin 3 were published, and they also found Kd values for pin 1 and 3 that were even higher and around 150 micromolar. So it appears that the substrate half maximal affinity for auxin binding is way, way, way above the physiological range. And this is also seen here when we do our oocyte experiment with pin 8. We again get the linear relationship. And the reason for this is obviously because in the physiological range, we're looking here at the very beginning when you have a seemingly linear correlation between the substrate concentration and the transport rate. So does that matter? So what you see here is the speed of the different pin and kinase combinations. And what you can see is that here, when you have a slow transport rate, you can reach a high auxin maximum. However, at the other end, when you have really quick auxin transport, a cell in which you have pin 3 and pinoid will probably never reach as high an auxin maximum as a cell in which you have pin 1 and D6. And for this to further investigate that, we asked Kirsten Tuscher to plug it into the model. And first, so this is the old model, which predicts fairly high auxin levels in the columella. And when she now puts in our experimentally derived parameters, what you can see is that we get, or she gets, considerably less auxin in the columella. And this now also fits much better here with the data that Karin Jung obtained when they looked at auxin levels here, and they found that the columella is pretty much devoid of auxin. So I wanted to talk a little bit more about the loop. And what we did is we performed a domain swapping approach. So in principle, we permutated all the transmembrane domains and all the loops between pin 1, 2, and 3, 3 as a representative of 3, 4, 7. And the readouts were transport rates in oocytes. And to make that very short, all of them were functional. So pins behave like modules, so and very, very well behaved with different properties. I'm going to talk about that in a little bit more detail. We also looked at localization and polarity of the constructs in epidermis cells, and we determined the vertical growth index when we expressed all these chimera under control of the PIN2 promoter in the PIN2 mutant. And the vertical growth index takes the length of the root and the root angle into consideration. And this has two components, a growth component and the angle component. Please remember this. So now when we look at the parents for the domain swapping approach, I mean, here is nothing new. When you look at the polarity index, the apical localization you find in pin 2, you don't see apical localization in pin 1 and pin 3. But then now the interesting thing is when you look at the vertical growth index, what you find is this is what you would expect when you express pin 2, you get it back to wild type levels. However, when you look at pin 1 and pin 3, they are somewhere in between. I mean, they're significantly different from wild type and they're significantly different from the mutant. And the reason for this is that the vertical growth index considers the angle and the growth rate. And it is known that the PIN2 mutant also has a growth defect. So it has shorter roots. So these here are biological replicates, each consists of 20 roots. And what you see, this is the growth rate of the wild type. This is the growth rate of the mutant. And all of them can complement the growth defect just fine. However, when you look here at the Rose diagrams, so the angle, there is only very, very little effect. But I want to stress the word little in this context because there is not no effect. So I, in the interest of time, have to focus on a few very, very interesting of these chimera. And the most interesting chimera are the ones that are the most extreme. So pin 2, if you remember, is a super slow transporter. Pin 3 is a decent transporter that gets something across the membrane. And when we have now here the pin 2 transmembrane domains with the pin 3 loop, that has slow transmembrane domains and it has a loop that doesn't have a pronounced kinase affinity. When you have pin 3 with the pin 2 loop, you have the fast transmembrane domains and you have a loop with a pronounced kinase asymmetry. So I would have predicted what we have is now we get here a slow transporter that responds to both kinases, and we get here a fast transporter that responds to pinoid, but not really well to D6PK. And what we got in both cases was something in the middle. The interesting part here is that this here is a gain of function. I mean, if something is slower, you can have also messed up the protein. But if something becomes quicker, it becomes really, really complicated to explain this. And this is a pattern. Now, and when you look at the vertical growth index, both of them complement almost as well as pin 2. And this also applies to the angle. And interestingly, both of them are also localizing polarly. And this is consistent with findings that Jirschi had when they looked at the co-evolution and localization of pin transmembrane and root domains, where they also found that there is something about the transmembrane and the loop domain that is brought about by the individual parts to the localization of the proteins. Now, for all these chimera, we found that they complemented the growth defect of the PIN2 mutant, but there is nothing that could be correlated somehow to the transport rates. The interesting part came when we correlated the transport rates here on the x-axis and the root angle. And what we found then is a very, very strong correlation with a coefficient of about 0.8 that indicated the following that's probably better viewed here in this depiction. Because what we have here now is, or what we have done here is, we ranked the transport rates that we established in oocytes. So this here is the highest transport and this here is the lowest transport rate. And then here on this side, we ranked the root angles that we found for the individual chimeras. And what you can see here now is that what I told you before, that there is a couple of things. If you introduce a loop of a slow pin into the pin 3 transmembrane context, you make the transporter slower. But we frequently observe that when you introduce the pin 3 loop into pin 1 or into pin 2, we can increase transport. And this is then also reflected by the root angle, where you can nicely see that when we have pin 3, the super quick transporter, that has the worst root angle, whereas pin 2 and pin 1 with the pin 2 loop, there is actually no difference between those, can complement the mutant to wild type levels. So what that tells us, we have an inverse correlation between the root angle and the transport capacity. And what I found super, super surprising is that the transport rates that I would have thought are mediated by the transmembrane domains only seem to be depending on the root and the loop domain as well. And this is probably best highlighted here when we make something that is completely artificial, and we introduce the pin 3 loop into the pin 8 transmembrane context. These here are the transport rates of pin 8. Remember, this does not require activation by kinases. So here we have auxin transport in pin 8. And when we then look here on the other side, this is the other parent of the synthetic chimera. We have pin 3, that's auto-inhibited. It's then here induced by D6PK and by pinoid. And interestingly, now when we introduce the auto-inhibitory loop into the pin 8 transmembrane context, we can auto-inhibit the pin 8 transmembrane domains. And when we then co-inject the kinases and we measure auxin transport, what you can see here, this here is statistically significant from pin 8. But this one here, the pin 838 chimera activated by pinoid, almost goes up to pin 3 levels, which indicates again that the introduction of a loop into a transmembrane context can increase the transport. And we don't understand this at all, how this happens at the moment. We also tested these chimeras. We did a few others as well in plants. The problem there is that they are not very well expressed, so you hardly see any fluorescence, and if you see, it's non-polar, and there is no complementation of any phenotype that we looked at so far. But I believe in terms of the biochemistry and the modularity and how that works on a mechanistical level with auto-inhibition and transport activation, this is fairly interesting. So I'm coming to the end of that, and I want to give you 3 take-home messages, which I find is the correct dosage for take-home messages after a complex talk. And that is, pins have distinct transport rates. So when you model, please also consider that there are differences in the speed of the transporter. Pin 1 and pin 2 being really, really slow, and pin 3 being between 8 and 10 times quicker. These transport rates matter. And in terms of gravitropic root growth, slow and steady does the job. You want slow and steady auxin flux in the epidermis cell, and then you get complementation of the phenotype and more gravitropic root growth. And again, for me, this is the most striking observation. The pin loop domain, which is intrinsically disordered, is required for transport regulation. That was not surprising, but it also contributes to the transport kinetics. And this is obviously something that we're very, very much interested in, to look at that a little bit further. And I want to acknowledge our funding, and I also want to acknowledge the people who did the work. So this is mainly the PhD work of Dorina Janacek. Lukas is doing mainly the electrophysiology on pin 8 and what we're doing in this context. And Sarah is with you in Japan. She has a poster, which is P12, where she takes our domain swapping approach to the next level. And I believe she will also be able to answer your questions if you have any. And we have a couple of collaborations here. And the most important one in the context of this particular paper is Kirsten Tentuscher, who modeled what I showed you and a few other things that I didn't show you. Jiří, who we collaborated with Matusz Glant in terms of protein localization, and my longtime collaborator Klaus with the kinases, and in Bernhard Pfister did proteomics and phosphoproteomics for us with the Xenopus levis oocytes. And with that, I'm done and would be happy to take your questions.
Thank you, Lee. Questions?
Hey, Uli. Thank you for a really interesting talk. I really like the take-home message number 2, which is slow and steady does the job. I guess if you think about...
I assume, that's Malcolm. Hi, Malcolm. I thought it was you, but I can only see you now.
All right. I guess you want the auxin to hang around in the elongation zone to have an inhibitory effect. And so it's a question of, it not going shootwards too fast. I mean, Tatsuaki showed very nicely how you get this transient inhibition within the first few cells of the transition zone. So I guess you want to make sure it stays there long enough to have an effect. That's like I guess a combination of PIN and AUX1 would do that. Either essentially delay, keep it there until it's had its effect, until the statolysts switch, and then it's released. So I mean, is that your thinking?
This is our thinking, and this is pretty much what we also write in the paper. And what I left out here is we also looked at auxin levels, and I mean, it's not really auxin levels per se, but we looked at R2D2, and exactly what you're describing happens. This is our hypothesis, yes.
Okay, great, thank you.
And okay, I have a question. Pin2 loop regulates the localization of the pins, right?
Yes.
And how is it's actually regulated? What recognize loop of the Pin2 and what recognize the pin loop, a Pin2 loop. What's recognized? What?
I mean, are you asking the question what proteins interact with the Pin2 loop to mediate the localization of the pins?
Yeah, exactly.
I have not looked at that, but I believe that if you ask Jiří, he might actually be able to give you a more detailed answer about the localization of pins in epidermal cells and the apical localization.
Yeah, okay.
So I'm going to ask the reverse question of what I said before, which is slow and steady is great for when you're moving auxin shootwards to the elongation zone, but I guess in the columella, you want something which is really quick,
Exactly, and this is also what's happening. Yeah,
exactly. So you want the Pin3 to be dynamic and respond.
Yeah.
So that's the current thinking, I guess.
Yes, and we will have to look at that in a little bit more detail, what happens when you do combinations. So when you take Pin3 out of the columella and you make something slow in there, and then you combine that with a quick transport in the epidermal cells.
Yeah, and I guess this comes back to Jiří's evolutionary paper, if he's paying attention. This is, I guess, faster root bending. Do you think that that's really driven through the evolution of Pin3 and Pin7? The fact you see faster bending, do you think that's the driver for evolution? That was what you were describing in the paper. Or do you think pin 2 is also part of this story?
I guess it's part of the story, and I believe that also Jiří has nicely demonstrated that the pin 2 loop also in evolution is required for a quick root gravitropism.
I tried to ignore my first calling, but when I'm called for the second time, I have to come. So,
Beetlejuice.
for the evolution story of the fast root gravitropism, just to explain to the rest. So we show that actually the fast root gravitropismus can be detected only in the seed plant, so both in gymnosperms and angiosperms. And that correlates with the evolution of the ability of pin 2 to be apically localized or shoot work localized in epidermis. But we never looked whether the pins 3 and pin 7 that are expressed in columella, whether there is a difference in the capacity or speed of the transport between the gymnosperm and angiosperm pin 3, pin 7 type proteins versus the older ones. So that is something maybe you can try, Uli, right? We just checking whether the Marchantia or Physcomitrella pin 3, 4 variant would not yet have this distinct property of transporting fast. This is something we never tested, so I cannot say. But yeah, it would make sense because it's also the anatomical feature of the statolids being specifically present only in columella evolves in gymnosperms. So before you actually don't need this rapid auxin fluxes in columella because the statolids are more broadly present in the whole root. I use the paper in my lectures, my teaching lectures.
I'm so honored, Malcolm, but we can exchange compliments later.
Hi, Roy. Sorry to say that, but I think you made a duplication in one of your figures. So I'm pretty sure it's a mistake, and I checked on I think it's on the figure where you show the localization of pin 3-2-3 versus pin 2-3-2. So yeah, this one there. So I don't know if it's.
Yo, I'm sorry.
Yeah,
I'm sorry.
So it was just so it doesn't get on the.
But it's not in the paper. It's here. It's not in the paper. It's here in the presentation. But thanks for pointing that out.
Yeah, short question.
So it's a short question. What exactly is at the molecular level that determines the rate? So you clearly show that the loop size is important. What actually is, in the first movie, you show clearly that it's a conformational change happens. So is that this turnover is faster with that loop, and it becomes slower in some kind of loop? So what the molecular mechanism behind this difference in the rate?
So if I understand your question correctly, you're asking me what my hypothesis is, why that when you have a loop that the turnaround is quicker.
Right. So what kind of the nature in the loop can change such the turnover rate of this the conformational changes?
There is no good answer because we don't know how that works on a mechanistical level. But if you ask me what my hypothesis is, I would say that it is that the introduction of a loop domain here is giving more degrees of freedom so that the transport can occur quicker. So somehow with the reduction of the loop size, it restricts how that transport is, how the flip back is working. I mean, this is mainly derived from the fact that the flip back, so turning into the inward open state, is the rate limiting step in many transporters, and it's particularly true for the pins. So I believe that the rate of the flip back is impacted by the presence of and identity of the loop.
Yeah, thank you. Yeah, is there any way to confirm that? Is it possible to kind of demonstrate experimentally that that's a cause for the turnover?
Well, I mean, the thing is, many people are trying to isolate these pins in with a loop and then determine the structural components. But I mean, I don't know how the progress is from the two Chinese labs, but our coworker in Denmark so far was unable to purify meaningful quantities, so we can get to that from a structural level.
Thank you very much.
Yeah.
Okay. Thanks, Uli.
Thank you.
The next speaker is Dr. Satoshi Naramoto from Hokkaido University. He will talk about evolutionary conserved mechanism of oxygen transport.
I'm Satoshi Naramoto from Hokkaido University. First of all, I would like to thank organizers for giving me a chance to present my recent works. Okay, let me start my presentations. As you know, the polar oxygen transport play an important role for the tropism and plant morphogenesis. Today, I would like to talk about the work about some evolution of the molecular mechanism of polar oxygen transport. My research question is that how has the polar oxygen transport system evolved, and what does the ancestral form of polar oxygen transport looks like? To address these questions, in my research group, we are analyzing the several different plant species to bryophytes, including Malacanthia polymorpha and Piscometриum patens. And also, we are also working on some monilophytes. But today, I would like to introduce my work of the Malacanthia polymorpha. In Malacanthia polymorpha, the genome structure is rather simple and single pin gene is present. First, I perform the genome editing and isolate the pin 1 mutant in Malacanthia polymorpha. And here is a growth. And this one is the top view, and this one is the side views. Maybe it's a bit difficult to see it, but in the side views, in the wild type, in the case of wild type, the rhizoid is developed from the ventral side, and green part is the dorsal side. And in the case of pin 1 mutants, this is the kind of side view, but it's a bit difficult to see. So actually, this phenotype is quite similar to this. Actually, this phenotype is reported in 1933, and he found that if you grow the Malacanthia gemma by using a clinostat, he found that the two leaf-like structure is fused, and it appears that the rhizoid is grows from both sides. And actually, in the case of pin 1 mutant, the phenotype is quite similar to this one. So therefore, I started to confirm these observations and check the effect of the gravity and some light by using the clinostat with some LED control systems. I irradiated the different light and under the microgravity conditions. When I grow a Malacanthia under the blue light and microgravity conditions, clinostat conditions, in this case, growth patterns looks like a wild type. So looks like wild type. And I also grow a Malacanthia under the white lights. I still couldn't confirm this phenotype. However, when I irradiate only the red light and clinostat, in this case, I could confirm the previous reports. So by comparing these two data, it suggests that the blue light is sufficient to determining these dorsal and ventral tissue patterning. And I further analyzed that the genome structure of Malacanthia in Malacanthus, in Arabidopsis, many pin regulators are already identified. I first checked the orthologs is present or not in Malacanthus. Of course, pin protein is present, and I also checked some membrane trafficking regulators, for example, the GNOME 1 BAN3, and some kinase D6PK and pinoid, and phototropin or NRL family proteins. I did a CRISPR knockout for all of them. Actually, the pinoid is not present in Malacanthia, but otherwise, most of them are present. I knocked out all of them, and I found that what I found is that first I analyzed the phenotype of GNOME. However, the GNOME mutant is a lethal, so it's not easy to analyze the morphology. However, in the case of BAN3, which encodes the alpha-gap proteins, which so kind of membrane trafficking regulators, in this case, I found that this mutant shows this is the image of the Malacanthia BAN3 mutant, and this is wild type, and this is the NPA treatment, so similar to the PIN1. So in this case, what I found is that in this mutant, the phenotype is quite similar to the PIN1. Moreover, I also analyzed the D6PK mutant, and I confirmed this type of phenotype. So for this, based on this data, it seems that the molecular mechanism of polar auxin transport is largely conserved between the bryophytes and seed plants. Maybe some of you feel that how this phenotype is interpreted. So let me introduce the morphology of the Malacanthia gemma. This is a developing gemma, and this is a mature gemma. And here is the dorsal side, and this is the ventral side. In the early stage, the gemma have a single apical stem cells. However, at the mature stage, actually there is a two apical stem cells. So what I'm thinking now is that it seems that the light signal most probably is the blue light and gravity. These two signals is important for determining the dorsal and tissue patterning. And in the case of the blue light and gravity is present, in this case, one of the apical stem cells localized at the upper side, but this apical stem cell is deactivated, and single apical stem cell is alive. And then this stem cell undergoes a multiple round of cell division and then differentiates the dorsal and ventral tissues. However, light and gravity signaling is compromised. In this case, these two apical stem cells remains also active, and then each of these apical stem cells undergoes a morphogenesis, and thus the two leaf-like structure is developed and fused at the dorsal side. So next I try to connect how does the auxin transport was involved in the dorsal and tissue patterning. So I established the functional Malacanthia polymorpha PIN1 GFP line and analyzed the subcellular localizations. As you can see here, the pin expression is accumulated around the apical notches. I could also detect a signal, stronger signal around the rhizoid cells. But when I observe this type of section, so vertical and transversal sections, here is the dorsal side and ventral side. In this case, what I found is that if the blue light and gravity if blue light is irradiated along these directions, in this case, it seems that the pin protein is less accumulated as compared to the ventral tissues. I also check the effect of the gravities, by using the red light condition, red light and 1G and microgravity conditions. In this case, I observe the dorsal tissue and this area is the apical stem cell areas. In this case, under the 1G condition, the signal is a bit weaker here. However, under the microgravity conditions, the signal around the dorsal part of the stem cell area is much stronger as compared to the 1G conditions. So it seems, and I also performed the similar experiments by using the blue light. And in the case of blue light, I could see that the signal disappears from this area. So these data suggest that the blue light is somehow removing the pin protein from these areas. And I also check the pin localization in the BAN3 mutant. This mutant was encoding alpha-cap and localized at plasma membrane and controlling the membrane trafficking. And in this mutant, in the dorsal area, the pin protein still remains. However, in the wild type, pin proteins, less pin proteins detected, since this protein is localized at plasma membrane. So I think that this pin protein is not removed from the plasma membrane in this mutant. So maybe this gene is acting to removing the pin protein from the dorsal side of dorsal areas. And since I also found some connection with the blue light, therefore I analyzed the NRL family proteins. In NRL family, there is 4 different groups. And in the Malacanthia, in each group, the single gene is present. So this means that there are 4 genes in the NRL family in Malacanthia polymorpha. And I knocked out all of them. At the beginning, the single mutant doesn't phenocopy the PIN1 mutant. However, when I make a triple mutant between the male NPH3 and RPT2, here is the image. And in this case, I found that these triple mutants show a similar phenotype as PIN1. So this data also suggesting that the blue light signaling is very important role for dorsal and ventral patterning. So altogether, here is a summary of my findings that if the blue light and gravity signaling is there, in this case, the pin protein at the dorsal side is removed based on the membrane trafficking and blue light effect. And this finding is quite similar to the seed plants, because if blue light is there, then in this case, the pin protein is somehow removed. So based on these findings that I would like to say that the basic mechanism of the polar auxin transport is already present in the last common ancestor of the Arabidopsis and Malacanthia polymorpha. And lastly, I would like to thank these colleagues and this grant. Thank you very much.
Thank you, Satoshi. Nice talk. Question? We have 2 minutes. Okay, let's start. I'm just curious about the imaging of PIN1, GFP. We could see the dot. Is that the chloroplast or signal of the pin? Yeah.
I think this is a chloroplast, but maybe this is autofluorescence because the expression level of the PIN1 is not so strong. So I have to irradiate excited by the strong laser. So that's why the chloroplast chlorophyll autofluorescence is detected.
Okay, thanks.
Thank you very much for this talk. Does the phototropin mutant also look similar?
In the case of Yeah, when I make a knockout mutant phototropin, in this case, growth itself looks like normal in terms of the dorsal and ventral tissue patterning. But when I grow a phototropin mutant under the microgravity conditions, the phenotype becomes the pin1. I could see the pin1-like phenotype.
Okay, so this is why you conclude that it's both a light and a gravity response. Because I was
yeah.
wondering whether it could be simply a light response, but
Yeah.
this data indicates it's both.
Yeah, growth. This data suggests gravity is also involved.
Thank you very much.
Although the blue light seems to be a dominant role.
Others? We have
For the crosstalk of the blue light and the gravity, if you give both blue light from upside and the downside, how will the development of the rhizoid?
Blue light on both sides.
Yeah, because you said that the blue light inhibited the rhizoid development of the upside cell, right?
Yeah. In the case of if I irradiate the blue light from both sides, the phenotype looks like a pin1. I don't know why, but maybe blue light is really the effect of. In this case, the gravity effect must determine the dorsal and ventral tissue patterning, but it seems that the blue light is more play a dominant role as compared to the gravity. So that's why if you eliminate the blue light,
okay.
I could see such a thing.
Okay, thank you.
Thank you, Satoshi. Very nice talk. We're moving on the next talk, last talk. So next speaker is Dr. Max Joss from University of Cambridge. We talk about the FRET sensor for the auxin.
Okay, is it talking now? Yeah, okay, cool. Unfortunately, it's already a bit past and I am losing my voice. So I will have to speak relatively slow compared to normal pace. So sorry if we go a bit further, but yeah. Like many of you, I've been working on auxin and tropisms for a couple of years doing my PhD. And one thing that always frustrated me is that we assume we know where auxin is, but actually we don't. We presume where auxin is, but we actually cannot directly measure it yet. So this is important because, well, as we know, auxin basically does a bit everything. In this specific case today, this week we are focused mainly on those two aspects, but auxin does everything. And we need to know where auxin is. So we have a few tools that are already available and that are amazing to actually do that. So I won't present GR5 or D2. Everyone use them. They're super easy to use and actually quite good. They respond to very low concentration of auxin. But also they're very good, they have limitations. And the main limitations that they have is relatively low spatial temporal resolution, which as we heard a few times is a very important aspect when we look at the dynamic. So there is other tools that have arrived on the market recently. So there is Auxin, which is the first direct FRET auxin biosensors. So auxin is a very, very interesting bioengineering story, and I really invite you to look at the paper. But although it is interesting, it has one main limitation, is actually the affinity of the sensor is limited. So it is outside of the endogenous range. So basically, we can sense auxin, but not in a meaningful manner. So this is exactly what I try to tackle by doing something, a sensor, in this case, a FRET sensor, that will have all of those characteristics. So we want something that is specific. We want to sense auxin, not IPYA, for example. We want something that is reversible because as we heard, auxin dynamic goes both ways, accumulations, but also depletion. We want a sensor that is going to work in the endogenous range. So what we think a real endogenous range is probably between 0.1 and 100 nanomolar. This is probably where we are actually. The fourth thing that we want with a Fred biosensor is that it has a good signal-to-noise ratio. So basically, I will explain you briefly what a Fred sensor is, but I will invite you to actually ask me questions at the poster because I don't have much time. But yeah, we want the signal change to be as big as possible because the bigger the change, the easier it is to quantify. And finally, when we stably express the sensor implants, we don't want phenotypes. So there's quite a few ways to prepare to engineer those sensors. I won't go into details, but basically I will tell you there's three parts. The first part is the sensory domain. So this is where the auxin is going to bind. There's the thread pair, which is the fluorophores that are doing the magic in a way. And then there's linkers, which is basically just short amino acids that are here to link all together. So what Kartika and Martin, which are also postdocs in Alexander Jones' team, did was for 7 years trying to engineer a thread biosensor. So what they did is they used a bacterial transcription factor, and I won't go so much into the details, but basically it's one slide for 5 years of work here. But basically they managed to make a first version of the sensor, which we called YAGO1, which is IAA genetically encoded optical biosensor. And what we can see here is basically when you add auxin in the system, you have a shift between the emission ratio and of the acceptor protein and the donor, right? So this is you have a signal change, what we call a ratio change, and this ratio change is ultimately what we measure. So to do this engineering, there's quite a few steps. It's also explained in the poster, so please ask me questions after, that will be easier. But basically, you can either directly do a structure-guided mutagenesis if you have the structure of your protein, or you can also do directed evolutions with random mutagenesis, for example, and screening. So once again, we skipped 2 years in time, and now we are in Planta. So we managed to stably express the first version and the second version of the sensor. And what we did is then we validate them in plants. So we use ubiquitin promoter. So what you observe here is every dot is a nucleus. So it's an NLS tag. And what we observe is this color coding basically represents this emission ratio. So when you have a color, when you have a color that is in the red, it means you have low auxin. And when you have a color that is in the blue, it means you have higher auxin because it's negative charge. Anyway, so what we did is you have the seedling Arabidopsis. If you add auxin in your system, so 10 micromolar for 10 minutes, you have a drop in the ratio. So you detect more or less auxin. And if you want to do the reverse, so if you want to block auxin biosynthesis or auxin transport, for example, you can also observe that you have a change in the ratio. So you can observe actually depletion or reduce amount of auxin in the system. So we are very pleased with that, but there is some limitations with those first two versions of the sensor. So the first one is we have a slight phenotype. So as I said, we use a bacterial transcription factor as the sensory domain, and a transcription factor might have an effect even if it's not directly a plant transcription factor. And we observe a root phenotype. We don't observe much phenotype later on, but still this is important because most of the data we saw last 2 days were on seedlings. So it is something to consider. And if I wrap up basically these 7 years of work that Martin, Kartika, and then I did, we managed to go all the way from this first version, which was the purple there. So we have a sensor with a good signal-to-noise ratio, but with not terrible, but with a very bad affinity. Basically, we could sense auxin, but not in a meaningful manner. From there, we managed to go to the blue line, which is the YAGO2, so the second version. And this second version, which is the one that is stably expressed in plants, has an affinity about 250 nanomolar, but the signal-to-noise ratio, as you can see, is relatively low, about 20%. And this is in vitro data. It means that once you look at the plants, it's probably less actually, because plants are noisy systems. So because of that, I managed to make a new version recently, which is called 2.1. So I already presented those data a couple of months ago, and I think some of you already have seen them. So you might be wondering, well, why have you not expressed it in plants yet? Well, there's a plot twist. Is because in my pocket, I had done screening for another sensor and actually managed to make another sensor, which I will present today. So those are primary data and unpublished, so please keep it for yourself. But yeah, so we did YAGO3. So what is YAGO3? It's basically a new design. It's a Fred biosensor as well, but it's on a new design. And not only we did one sensor, but actually we did 5. Well, I'm presenting 4 here, but there's 5. So we did 2 sensors that are called NR, which are non-responsive. Those are negative control. Then we made the LA, which is a low affinity version. So this is the purple here, which has a KD that is written there. Then we have a medium affinity version. which is that has a KD of 450, 300 to 450, let's say. And then we have YAGO 3 high affinity. So YAGO 3 high affinity has a KD that is between 30 and 50 nanomolar, and the rate of charge is above 100%. So basically, this is the story of the engineering from YAGO 1 to YAGO 2 to YAGO 3s, with the last version being the green one here. So that looks super promising. This is in vitro. So that looks super promising. So the first thing we wanted to know is, are we sensing oxygen with that? So what I did is doing a screening for specificity, so against different oxygen-like compounds. And what can strikes is that 100 nanomolar is already a big peak here. So we also did the titration, and you can see it's highly specific for oxygen. So not only it is specific and with high affinity, but it's also fully reversible. So what I did here is basically purifying the protein and then doing a buffer exchange. So basically we remove small molecules from the protein, and then you spike them again. And we can see that when you do the buffer exchange, you can clean the sensor, which indicates reversibility, probably full reversibility actually. And any good story has some drawbacks. So although it is a very good tool, it is pH sensitive. And the reason for that is because I use a YFP as one of the fluorophores, and YFP has a pK of 5.8. So the sensor works very well at pH 8, 7, and 6. But then when you look at pH 5 here, you can see that you have a massive reduction in terms of signal change. It's simply because the YFP is getting oxidized, probably, or just is sensitive to the acidity. And if you go at pH 4, then you basically completely kill your YFP. So you can't have threat anymore, simply. So that's in vitro. So I did quite a lot of assays to make sure that I was not going to present crap data. And this is quite consistent. So I already did all the experiments for the paper. Now, what about plants? Does it work in plants? Well, that's a big question. I don't know if you saw the date that was written on one of the plots earlier, but I basically finished that last month. It means I don't have stable plants yet. But because I don't have stable plants, I thought, okay, I will do some transient tobacco. So at least I can please the audience. So this is what we have here. So this is, once again, nuclear localized version of this YAGO 3 high affinity sensor. So what we did is transient tobacco, and then I spike my tobacco with oxygen, and I measure again the emission ratio. So you have the high affinity here. So you can see that 3 biological replicates. You have a consistent increase of emission after oxygen treatment. And the NR, which is the non-responsive control, you don't have changes. So it's indicate that it probably works very well in tobacco. And not only does it work with a nuclear version, but it also works with a plasma membrane version, which I think is going to be very interesting in the future. So this is exactly the same I'm showing you here. So this is the high affinity in the plasma membrane on the cytosol side using a finalization domain. So this is before and after oxygen treatments. And this is the NR as a control. And what I'm showing you on the side here is the 3 channels that we use when we do the microscopy. So we measure the CFP alone, the YFP alone, and then the FRET, where we excite CFP and measure YFP. And the last one is basically just the analysis output. So this is just a color coding number, coded number. So what do we have so far? So we started with the red, YAGO 1. We managed to go all the way to the green, YAGO 2, which was improved affinity, but lower rate of change. And all of a sudden, we managed to go all the way to this guy here, which is YAGO 3. This is a question that I had a few times today, so I did ask it, I did put that on my slide. This is probably the physiological range in Arabidopsis. So although we can still try to go a bit further down with very low affinity, with very good affinity, I think we can already probably sense oxygen in planta. So the next step that I have is do stable Arabidopsis, engineering a pH tolerant version, and then do what I am paid to do, which is to actually do plant biology and answer some questions. So thank you all for your attention. Thanks to the Jones team, amazing people, great to work with them. If by any chance anyone has this line, that would be great. Pin 3 with the RFP. The RFP is important because those are channels already picked by the sensor. So yeah, that would be great. Thanks.
Thank you, Max. It's a fantastic talk. Question.
That's really great. It's going to make a big difference. I mean, one thing I guess you're worried about is phenotypes. Do you have a sense of binding affinity, whether it will be hoovering up the auxin, interfering with the endogenous levels? Do you have any sense of that?
That's actually a very good question. There's a good point that already, I think Dolph asked me that last time I presented, is it not going to trap auxin? Well, at least in vitro, it is fully reversible. So it's very likely that it's not just a one-way binding. It just binds and it stays bound. I think auxin will be able to just flow through, but it needs to be validated. And yeah, if it binds auxin in a reversible manner, then yes, there probably will be some phenotypes which would be an issue, and then it means more engineering needed. Yeah.
Yes. Yeah, first of all, congratulations to make a very nice tattoo. I have a question. Have you tried a sensor in the plant growth response with improved gravitability?
You mean for the last version is not in Arabidopsis yet, but for the previous version, yeah, I tried. The signal to noise ratio is very low, which means that it's a very system. Hello. Okay, it's a very noisy system once you express it in Arabidopsis. Also, you can't really go too deep in the tissues because it's a dim, the sensor is relatively dim. So it's quite hard to get this nice focus plane of the classic DR5 pictures, and yeah, so far now I could not actually observe gravitropism induced auxin redistribution.
Thanks.
Yeah, I guess talking to Xander, and the hope is to have these biosensors going to different compartments, yeah, and that would be just a game changer. So given the pH sensitivities, which compartments do you think you could use using the conventional one, and how could you address the pH shortcomings to actually then look at the apoplast, for example?
That's exactly where I sit at the moment. So since I finished engineering of this one, I spent the last 2 months just trying to tackle this pH issue, because that's exactly what I want to do, put it in the apoplast, because there's probably a very big pool of IAA in the apoplast. There's quite a few ways to do that, and I'm very happy to discuss it at the poster, maybe, because I think I'm out of time.
What's that?
If I have 1 more minute, I can answer the question. Well, yeah, so if you want to tackle the pH tolerance, you can change the Fred pair. So yeah, I'm using a YFP. I could decide to use GFP CFP instead, or BFP GFP. We know the GFPs are a bit more pH tolerant compared to the YFP, so that's an option.
Cool. Thanks, Max. We'd like to thank all the speakers again.
So, okay, this all session is finished. So, and I have 2 announcements. First one is, if you wish to go back to the Okazaki New Grand Hotel,